Hi, everyone. We're just going to give it a couple of minutes for folks to log on as they connect to Zoom. Okay, we will go ahead and get started. So thank you all for joining us at the webinar. So we have two great presentations lined up today, 3 speakers. So first we'll be starting with Christine Stauber at Georgia State University, and she's going to be presenting on arbovirus surveillance in wastewater. And then we have Kyle Bibby and William Chen from the University of Notre Dame, and they'll be presenting on modeling tools to evaluate wastewater-based surveillance targets. Just a couple of reminders about ongoing RCN activities. So we have our monthly journal clubs. We are always accepting webinar nominations for people to present here. And then we have an email list, and I will send out links for both the nomination form and the email list after these intro presentations. We have an upcoming workshop. The participants have been selected for this. So we will keep an eye out for the recording of that once it gets posted on the website with the summary. And then just a brief note, the January webinar is canceled. We realized that this was still during a lot of individuals' winter breaks for their universities. So to avoid low participants and scheduling conflicts, we just opted to cancel it. So we will look forward to seeing you all at the next webinar in February after today. So I will stop sharing. Christine, you can go ahead and get ready to share your slides.
Just a reminder, as Christine's sharing, to put questions in the Q&A, and we will be moderating, Sarah will be moderating the questions at the end.
Okay, great. Well, first of all, just thank you for the opportunity to present at the webinar. I am looking forward to sharing a little bit about my work here, and certainly providing this at a very high level. But my talk today is titled Arbovirus Surveillance in Wastewater from Proof of Concept to Implementation. And most of the work that I'll be talking about today is really on the proof of concept end. So just a brief outline of what I hope to share with you today. I'll just give a brief rationale for why we might want to think about using wastewater surveillance for mosquito-borne arboviruses. Those are the type of arboviruses I'm going to focus on today. I'll talk about some work we've been doing with Zika virus, and then a little bit of preliminary work we've been doing with dengue virus. I'm going to focus my talk on applications in Brazil, and then talk a little bit about the potential to use implementation frameworks, ethical considerations, and acknowledgement of all my collaborators. So let me just... Why arboviruses? Maybe I don't have to convince anyone here, but arboviruses are an important human group of viruses that cause human disease. They are RNA viruses transmitted to human and other vertebrates through an invertebrate host. And of course, prominent human pathogens from this group include a variety of mosquito-borne viruses such as Zika virus, dengue virus, West Nile virus, and yellow fever virus. These are all flaviviruses, but of course there are others that are not in the flavivirus family. Infection may result in a range of symptoms from mild to severe, but in many cases, these diseases are asymptomatic. Zika virus, dengue virus, and other arboviruses are transmitted by the common mosquito vector Aedes, and in most environments, they are often co-circulating. And as we know, there's a shifting range of disease transmission of these mosquito-borne viruses due to climate change. As highlighted in this article by Dickens et al. In 2018, the suitability for these two vectors, in particular Aedes aegypti and Aedes albopictus, is quite broad. And what we know is that from this work, that accessibility, which is really a measure of humans interacting with the vector, absolute humidity and annual minimum temperatures are very strong predictors of the potential presence for these vectors. And then again, the concern. We've seen local transmission in the United States multiple times this year, and the Americas is experiencing some of the highest number of cases they've seen for things like dengue virus. So that's a growing concern. I want to focus the beginning of my talk on the work that we did because of the Zika virus outbreak in Brazil. We were in Brazil at the time that this started happening. Colleagues, of course, were working on a number of projects, but in 2015, Brazil notified the World Health Organization of a mysterious illness circulating throughout its northeastern states. In the first 5 months of that year, they identified at least 7,000 cases of illness with a skin rash, which was sort of a unique feature. But beyond that, the symptoms were quite similar to other arboviruses. Over 400 samples were taken, but few were positive for dengue or chikungunya. And in May 2015, Brazil's National Reference Laboratory confirmed that Zika virus is circulating in the country, which is the first report of locally acquired Zika virus disease in the Americas. Of course, initially, everyone thought this was just a mild illness until we started to see the major impacts, which of course are, congenital impacts in children. So we identified an opportunity to start thinking about whether or not wastewater could be used for surveillance for Zika virus in collaboration with colleagues in Brazil. As you know, the majority of cases are asymptomatic. And in the context of Brazil, and particularly northeast region of Brazil, people experience febrile illness frequently and often don't seek care. During this time period, my colleague Federico Costa and his collaborators had been and are still doing comprehensive cohort studies to understand the distribution and environmental and other risk factors for leptospirosis. And they were doing this work during the Zika virus outbreak. And they had collected samples for environmental waters in the community. And you can see these are pictures from the community that they are working in called Pau de Lima. It's a dense population, very hilly, and often lacks adequate sanitation services. So our preliminary work with Zika virus, which really started back in 2016 or 2017, was focused on our central hypothesis that we could find Zika virus in urine and therefore potentially in sewage, and this might enable detection of community level infection. And we started this work with just a simple proof of concept experiment to demonstrate to both us and others that RNA from a single-stranded RNA flavivirus would persist in environmental waters. I feel like nowadays this concept is not that odd, but maybe 5 or 6 years ago it was kind of considered like, why would these persist in environmental waters? And so that was really the beginning of our work. We then set out to do a little bit more comprehensive understanding of this, where we examined 3 conditions for Zika virus and sewage, where we looked at 3 temperatures we thought might be reasonable to find in places like Salvador. It could potentially get that hot there at 35 degrees Celsius, and set out to demonstrate that, under these temperatures, Zika virus RNA is more environmentally stable than we assumed. This initial study provided rationale and support to further explore materials and methods for the detection of Zika virus RNA in wastewater. And at the time, it also provided us some confidence that we could process these archive samples because we were able to, in pilot studies, extract RNA from spiked pellets. And just to clarify, we didn't have a chance or control over how the environmental samples were captured in Brazil. They had been, tested for leptospirosis, which was a simple centrifugation method of environmental waters. And so we had to go along with that method, which is one of the things that we examined in our next set of studies. So after our initial study, understanding whether or not Zika virus RNA would persist, we went to do a more expansive study where we looked at environmental water sources, virus concentration, time, and temperature as how they would impact Zika virus RNA persistence. We also looked at using skim milk flocculation as a concentration method. We compared nucleic acid extraction kits and the impact of freeze thaw on these samples. We analyzed the archive environmental samples from Brazil, and separately in work outside of this wastewater research paper, we analyzed Zika virus RNA sorption method using Morse swab. The rationale for the work was really based on two things. The first was that we knew in the communities that my colleagues are working in Brazil, there's often a mix of wastewater and surface water. Many of these communities don't have network sewers or partially networked sewers. And so we know that the surface waters were often part of a wastewater matrix in these communities. So we set out to understand both the relationship between just turbid surface water itself and then a combination of surface water and wastewater. We looked at environmental conditions to understand the potential for persistence. And then we started to look at, could we potentially recover the virus from waters using, simple concentration methods. The overall findings from that work, perhaps not surprisingly, suggest that increasing temperatures reduced persistence in both surface water and wastewater, although it was more pronounced in surface water. Surface water was also an important predictor of persistence. So all of our conditions suggested that the presence of surface water, especially alone and not in the presence of wastewater, was going to result in more rapid degradation of the RNA signal. We also looked at whether or not initial viral concentrations impacted persistence, and we saw that decay rates increased when we lowered the virus concentration of these seeded experiments. And we found that recovery using skim milk flocculation was lower in surface water than in wastewater. But ultimately, I think from the results, we think that skim milk flocculation and Morse swab, which I didn't show the results of here, do warrant further consideration for application in the field. I'm, still enthusiastic about this work, but I have really thought about how do we take this to a disease that, of course, is, circulating and increasingly of concern. And as I mentioned earlier, dengue virus is one of the most important emerging or re-emerging arboviruses globally. The growing incidence in the past 15 years globally, but also this has been a prominent virus in Brazil for over 40 years. And currently, the only types of treatment are symptomatic and supportive management. In the settings where this disease is endemic, laboratory diagnosis can be limited due to the quantity of facilities, challenges with sample collection, characterization of serotypes, and potential cross-reactivity with other serotypes and other flaviviruses. So we think that there are important tools that wastewater could provide that are outside of the clinical setting. Currently, what we know about use of wastewater surveillance for dengue virus is that we have seen dengue virus RNA in urine of infected individuals. Similar to the work we've done, others have shown that, RNA from dengue virus does not decay extremely rapidly in wastewater, although current published papers about wastewater treatment plant detection of dengue virus had found no levels. There are two preprints that have suggested that we may be able to detect dengue virus in wastewater, which are exciting opportunities to really explore this further. From my point of view, where I wanted to start with our work on dengue virus has been primarily understanding excretion in infected patients. And this work is in collaboration with Guilherme Ribeiro at Osvaldo Cruz Institute, we were able to obtain data from a community-based enhanced surveillance study of patients with acute febrile illness and rash in Brazil, in the city of Salvador. And you can see here in this diagram the community of Pau de Lima, which I mentioned earlier. This figure is from a publication looking at dengue acute febrile illness cases per population in this community-based enhanced surveillance study. In our work currently, which is where the aim was to investigate the presence of dengue virus RNA in various matrices of infected individuals. This study did collect a total of over 4,000 patients who were recruited and had samples collected. And of these, 96 were confirmed positive for dengue virus via dengue infection via serum lab test. We then tested urine and saliva for patients that had tested positive, and we detected dengue virus RNA in urine in less than 10% of these samples. This work is preliminary, but we're hoping to get a draft publication out soon. So I'm going to sort of transition from where I'm working as really proof of concept to think more broadly about arbovirus surveillance. I think we've shown in the United States and other places, primarily in high-income countries, that wastewater surveillance is promising and it can enable prompt public health action. There was recently a paper published in MMWR, and certainly the National Academy of Science, Engineering, Medicine has been writing reports in collaboration with organizations like the CDC. But my question is, what would this look like for viruses like dengue or Zika virus in Brazil? And I think there are some challenges and, of course, some opportunities for this. In Brazil, most communities have network and non-network sanitation conditions. So we need to consider what that means in terms of sample. And of course, those who are made most vulnerable to viral infections like arboviruses are unlikely to have network sewer. Separately, for dengue virus, really would like to understand the role of immunity and what that means for shedding on viral detection in those kind of matrices. In our particular small sample, we know that detection of RNA in urine was much lower than we've seen in one or two other publications in the literature. And it's unclear what that means for a place like Salvador or Brazil, where you see episodic events for dengue every 3 to 5 years. And separately, what is the recommended public health action? Unlike our current respiratory viruses that were been the focus of a lot of the wastewater surveillance in the United States, there is no vaccine that you can deploy when you detect these. And so really thinking about what it means and how you take public health action. Is it dispersal of resources? Is it more dedicated testing? But separately, I've been working in Brazil for close to 10 years now as a faculty member, and there's strong laboratory and diagnostic capabilities throughout the country, which may enable adoption of these wastewater surveillance tools on a more systematic level. And the country also has a universal health system, which can potentially help comprehensive data systems that may facilitate linkage to clinical disease trends. Separately, I wanted to suggest that as the field of wastewater surveillance moves forward, that perhaps we think about an implementation science approach. Traditionally, implementation science has been used for interventions in clinical settings, but it's growing in its application in water sanitation and hygiene. And I think just thinking about the tools, implementation science can provide a systematic approach for developing, evaluating, and bringing evidence-based interventions to scale. And so, as the network is growing to implement this as a public health tool for action, it would be important to start thinking about the ways we can use implementation science tools to examine, both process and impact. Even just using a simple implementation framework that's commonly used, like the REAIM framework, we're thinking about, these different categories of this intervention of wastewater surveillance and how it can impact all of these different features. Lastly, I'll just say that, as this field has grown, there's also growing understanding of the potential ethical questions that remain. Perhaps early on, I was very enthusiastic about this being sort of an aggregate urine sample, but as the field has matured, we know that there are questions around data collection, data analysis, data information systems. Of course, we would, hope that the use of these systems could advance and protect public health, but we have to be thoughtful about how we do that. And, I just give a couple of examples where, perhaps with the use of mpox detection in wastewater and illicit drug use, we can see where this type of tool could be generating stigma or result in potentially increased presence of police or authorities in communities that are, historically targeted for or, stigmatized. And so I strongly encourage, as this work moves forward, for, me and others to think about communities and stakeholders who can guide this work and help us assess the equity of the impact. And, recently there's been articles to suggest that structured ethical review, similar to an institutional review board, could be considered. And then lastly, I just want to say that, while sampling of wastewater provides a collective community sample, concerns remain for those who have been historically excluded from the decision-making process, as well as access to the services of healthcare and water and sanitation. And these questions are even more relevant in locations which contend with substantial inequality and limited resources, which we find both in the United States and in other countries. Okay, so with that, I'll just mention, this work wasn't possible without my key collaborators. I've mentioned a few of them in my talk. I've listed a number of them here. This really has been a wonderfully collaborative effort. I'm super grateful to all the support of my key collaborators and all of the students who have worked on these projects. And, here are a couple lists of the funding sources, but of course, as the network extends, they're more extensive than this. And so lastly, I'll just leave you with a picture of the lighthouse in the tip of the peninsula of Salvador, and just hope that maybe what I've shared today was a little bit illuminating about how I'm thinking about this work, and open to any questions, and hope I kept to time.
Yeah, thank you. That was really interesting. We do have some questions. So there's a few about how the different types of contributions might affect detection. So both if there are any environmental contributions, as well as from mosquitoes, like, are you able to differentiate between what would be coming from infected humans versus the environment versus mosquitoes?
Yeah, so in our work, we haven't been doing that. In the work with dengue virus, we have been collaborating, our initial work with dengue virus, we've been collaborating with Uriel Catron, who's at Emory, who has really done some work on this. So the short answer is, I don't know, but I'm not sure, our discussion around that has been, it's been hard to find this in mosquitoes. So I'm not extremely concerned about it as sort of overshadowing the human contribution.
Great. And then there's a question about how this might be applied in potentially non-endemic settings as dengue virus expands its regions. If there's a difference between testing with infected travelers versus, finding where it's recently entered new communities. Do you have any thoughts on that?
Well, I don't immediately. I certainly think the context of Brazil is very different than the United States, but there is a preprint and potential, I think, upcoming publication about this work in the United States that was able to sort of think about this, I'm not sure that you can distinguish when you see the signal in wastewater yet, particularly who's the source of it. Certainly in, if I think another analogy might also be sexual transmission of Zika virus in the United States was a prominent concern, whereas in a place where it's endemic and, arboviral disease, mosquito-borne transmission is endemic, it's not a prominent concern. And so those are sort of some of the things I'm thinking about. But I'll just say that there is a preprint out there that could perhaps provide more answers than I have about that.
Yeah. And then there's a question as well about whether or not these viruses are in feces or other body excretions on top of urine, and if you have any experience with testing other types of samples from humans, or if it's just urine.
Yeah, I mean, I think, most of the work that we've done has been focused on, the very preliminary work with dengue virus has been on urine and saliva, and it is present in those with dengue virus. I think we see, and we didn't do the work with Zika virus, but it's present in much higher numbers in Zika virus. And I think there's some small evidence that it's in feces of Zika virus, but Zika virus was everywhere in the body. It crossed the placenta, it was in the brain. I don't think we see that with dengue virus, so I'm not sure where it might be. And so I think those are questions, I think it's much harder to get fecal samples from individuals. And so it's not the typical sampling tool for these diseases in endemic areas. So I think it's important, but we don't have a good answer on it yet.
And then there's also a question about how the mixing of surface water and wastewater might dilute out the pathogen signal. Do you have any data on relative fractions? I think also is, maybe you could comment on some of the climate of Bahia, if it's affected by the seasonal rains of the Amazon. I'm not sure if that would affect detection in this setting.
Yeah, I think there is some question about the potential for dilution. I think there's maybe two questions. Does rainfall and then surface water, runoff influence the overall concentration, but does it also increase the likelihood that degradation of the RNA signal is more rapid? I think in many places in the world, we will see sort of two systems, right? You either have a system that's not conveyed by water, where you might have higher concentration of the waste, or you have ones that are these sort of open mixtures of both surface water and wastewater. And so I think we need to address this more broadly as we start to move beyond just the traditional wastewater systems we see in the United States. And again, I think, I'm just going to continue to advocate that Brazil is an important country to be thinking about this work.
And then was any data collected on the biochemical nature of the wastewater? Would that affect detection of these viruses at all?
So our work with the seeded samples, we didn't do a lot of measures beyond what the traditional wastewater treatment plant might measure. I think, the biggest predictor in that paper was water type and temperature. And so I think, more could be done to understand this. But in general, I think there's enough robust information that, yes, it might vary, but I'm not sure that it's going to vary enough not to make it worthwhile to do.
Great. Thank you so much. Thank you everyone for questions. That was really, really interesting. So now I will hand it off to Allie and Kyle and William.
Okay, William and Kyle, you can go ahead and share your screen.
All right. There we go. All right. Well, I guess as a participant in this RCN, it's really exciting to get the opportunity to present some of our work. And we're actually going to sort of dovetail very nice with Christine's talk with a little bit of modeling of Zika virus, but we're going to be talking about some of our Williams effort on developing modeling tools to evaluate and wastewater-based surveillance targets. So first, of course, giving credit to our great team, and the work that we'll be presenting today is mostly Williams' PhD work. So I think everybody here is familiar with the idea of wastewater-based epidemiology or wastewater-based surveillance that has gotten a lot of attention in response to COVID-19. The idea that individuals infected with the disease might excrete that pathogen into wastewater through feces or urine or saliva, and we can measure it in wastewater and then better understand what to do in response to that disease. And we also have known for quite some time that there are a very large array of pathogens that we find the signal of in wastewater. So this is some metagenomics work from about 10 years ago from my PhD, actually. And so I think the combination of those have led us as a community to think about developing wastewater-based epidemiology for a whole wide array of targets. And we begin thinking, oh, well, what's seen in wastewater, so therefore it's suitable for wastewater-based epidemiology. And I think that what we're finding as we work to develop this is that really some of these targets or wastewater-based surveillance is not one size fits all for these targets. And so if you think about the process of developing and evaluating and implementing a wastewater-based surveillance target, I think we as a community have spent a lot of time thinking about sampling methods and concentration and processing methods and extraction and appropriate molecular approaches, and probably have spent a little bit less time thinking about shedding dynamics, disease dynamics, water use in the community that would all impact the suitability of a target for wastewater-based surveillance. And so our modeling approach that we are working to develop for many different targets really begins to think a little bit more quantitatively about disease dynamics and shedding and what that means for the suitability of that target for wastewater-based surveillance, and also the potential benefits that we might gain from improving some of these analytical methods, whether or not it's necessary or is there some critical threshold. So with that, I'll hand it over to William to talk a little bit more about this model and model development.
All right, so as Kyle touched upon wastewater-based epidemiology, though promising, has some drawbacks. One being that there are many uncertainties. For example, it's unclear why at certain situations it works and in other situations it's not successful. Therefore, when researchers simply conduct experiments right away without a clear understanding of the why, they can basically they're essentially playing a guessing game on whether or not it would their efforts would even be fruitful. Therefore, to address this issue, we've developed a model-based WBE feasibility analysis framework that can help clarify what exactly affects and influences the likelihood of successful wastewater detection, and therefore help inform researchers on how to better think of approaching this technique to certain targets in certain situations. So here in this diagram is the overall workflow of the framework that we developed. Overall, we use input values as shown on the left to make predictions about output values shown on the right. And this model can be applied to any given disease target, so long as the input values shown on the left are available. So the first step is to conduct literature review on shedding dynamics of the disease target of interest, we then divide that by the wastewater per capita flow rate to be able to estimate a distribution for the pathogenic genome wastewater concentration for a particular location. It could be as broad as a country or as narrow as a specific wastewater treatment plant. So the next step then is to do the same thing for the process limit of detection, or PLOD, which I define as the assay limit of detection of the PCR assay and normalize back into the wastewater sample volume, which accounts for all DNA and RNA losses throughout the WB workflow. So essentially, this is a very important metric that is one value summarizing many important laboratory factors like the level of concentration, the process efficiency, and of course, the sensitivity of the PCR assay. So with both of these data, we can then be able to do the next step, which is to actually find the probability of wastewater detection. So as shown here, for a single PCR reaction, you could think of the probability of detection as the fraction of this wastewater distribution of the concentration of the DNA or RNA that's above a certain PLOD value. For multiple PCR replicates, we can also mathematically determine the probability of detection. Now, once we have all this information for a particular location and particular laboratory methodology, we can be able to construct probability versus infection rate curves that can help us predict the minimum infection rate required to achieve a certain level of detection, for example, 50% or 90%. So using the criteria that we want case rates to be above this minimum infection requirement, we can compare this rate to reported case rates. This will help us understand what the optimal PCR replicate would be at a particular PLOD value. This is important because the more PCR replicates you use, the more time and resources you would be spending. Therefore, depending on how relatively high this PCR replicate value is, you could have a better understanding of assessing how likely and how feasible WBE would be when applied to a specific disease at a specific setting and specific time. So here I summarize the 5 major factors which influence the level of detection for wastewater. And so the significance of our model is that it not only clarifies what these 5 factors are, but it helps quantitatively relate these factors to the probability of detection and also to each other. Therefore, researchers can answer important questions like, to what extent of an outbreak is needed for a given target to be detectable in wastewater, and perhaps more importantly, specifically how they should conduct their methodology. For example, how much more sensitive the PCR assay should be, or how much more they need to concentrate down the wastewater sample in order to achieve successful detection. I would like to also add a potential 6th additional factor would be the decay rate, depending on how significant it is. So overall, our model framework is useful because it can help identify which disease targets are suitable for WB applications, and therefore where resources should be allocated. And also it can clarify when, where, and how WB should be applied to be most successful. So when, meaning like how significant of an outbreak, where, meaning based on the wastewater flow rate, as well as based on the relative case rates in a specific location versus somewhere else. And of course, as I touched based on already, how and how much you should apply the technique. So here we'll sort of demonstrate the approach that we use for our model to two novel disease targets. So the first one is Mpox, which last year there was an outbreak occurring in many non-endemic countries, including in the United States and in European countries. Again, the first step would be to gather up from available literature the Mpox DNA shedding data from excreta such as saliva, stool, and urine. And from the results shown here, you can see that stool is the dominant shedding pathway for Mpox into the sewage system, accounting for about 7.8 log 10 genome copies of Mpox DNA per day. Now, dividing this by the specific wastewater locations per capita wastewater flow rate, which, for example, shown here for the average US wastewater treatment plant, we can construct a probability of detection versus infection rate curve at any given PLOD, which are shown in color-coded curves, as well as PCR replicate values. So evident from these curves, you can see that if you increase the infection rate, if you decrease the PLOD, and or if you increase the PCR replicate value, you would expect the probability of detection to increase. Also useful from these plots is that you can find what this minimum infection rate value would be needed in order to achieve a certain probability of detection. For example, 50%. So here are the results for the United States as well as for other countries. And I would like to highlight that countries with lower per capita wastewater flow, especially like Nigeria and DRC, are the countries that have the lowest minimum infection requirement, and therefore you will expect Mpox to be detectable more easily in these locations than in other areas like the US. So finally, we compare these minimum infection requirement data to reported case rates, for example, during the 2022 outbreak for the 5 countries examined here. And we can construct the optimal PCR replicate versus PLOD plots. And for example, for the DRC, if we use a pretty standard log 10 PLOD value of around, say, 2 to 2.5, you will need roughly 2 to 4 PCR replicates to expect 50% detection rate, which means generally Mpox WB applications would be feasible in the DRC and also in most of the other locations here, with the exception of the US, where you will either need to use over 8 replicates or consider increasing the sensitivity to a value of about a log 10 of 1 GC per liter. However, if you go to a hotspot in the US where Mpox had high case rates, the situation is now different, where now the plot is now shifted to the right, and you can see now using the same PLOD value of around 2, only 2 to 3 replicates would be needed. So overall, our model provides theoretical evidence that Mpox WB applications could be successful and feasible. And indeed, after we published this data, other papers have recorded and reported their successful detection of Mpox in wastewater at various locations around the world, including France, Spain, the US, and even in Thailand. So this is a good example of how our model predicted a successful WB outcome before it actually happened.
Great, thanks, William. And I'll quickly talk about another case that overlaps nicely with Christine's talk, and I won't go into as much depth because William did such a nice job of explaining the model. But you could see her for Zika virus, as Christine had noted. If we look at the shedding is dominated by urine as opposed to feces. And if we look at total shedding, it gives us a hint as to the suitability of Zika virus for wastewater-based surveillance, as we have about a 10-fold decrease in shedding for a Zika virus infection as compared to a monkeypox infection of only 6.7 log 10 genome copies per day. And we could see here, again, looking at the number of replicates that might be necessary in the US versus a PLOD, that we are getting up to very high numbers of PCR replicates that would be required for successful detection, right? So if we're looking at average case rates, even with a very low PLOD, perhaps infeasible PLOD of 10 genome copies per liter, you would need something like 10 PCR replicates for a 50% probability of detection. And in Brazil, at an average case rate, looking at a more reasonable PLOD of about 100 genome copies per liter, we would need maybe 4 or more PCR replicates. Now, what's interesting about Zika is that the case rates that we're using to estimate, especially during an outbreak, are guessed to be a 10 to 100-fold lower than the actual case rates. And so if we say that it looks challenging to implement this during a non-outbreak scenario, but if we look at an outbreak scenario, right, it actually suggests that we would move maybe significantly further towards feasibility because of the higher overall case rates. So this modeling suggests that it would be challenging to implement Zika virus WBE in a non-outbreak scenario, but it's potentially feasible during an outbreak. And so in closing, I think I would just like to say that I know we're all very excited about wastewater-based epidemiology, wastewater-based surveillance, but it really isn't one size fits all. And hopefully this modeling approach provides some framework to evaluate some of these targets and implementation. So targets for development, where to use it, when to use it, how to use it, and do that a little bit more thoughtfully. Of course, these models are only as good as the data that we have to put in. And I think a lot of folks would recognize that the data we have on pathogen shedding and also some pathogen dynamics, including persistence within some of these systems, is limited. And so we definitely need more shedding data to improve some of these modeling predictions. And there's always the possibility that some of these models could be wrong based on the shedding data that we have. But so far, it seemed to be a very powerful approach. So with that, we'd be glad to take any questions, and feel free to reach out to our group if you have any further questions after the Q&A today.
Thank you. Thank you, Kyle and William, for that really interesting talk. And it's great that you guys are working on the modeling side, because I agree with how Kyle started, that a lot of us have been working on measurements and not thinking as much about modeling, but that's super important and something that as engineers, we can definitely contribute. So there are a few questions, and I encourage you, if you have others in the audience, please add them to the Q&A. I'll start off with some questions about shedding. So can your model consider changes in the longitudinal pattern of shedding? And when you say shedding, in your models, do you use one number or do you use like a shape of the shedding profile, and can your model account for that, or is it meant to?
Yes, but I'll do the first question. You can do the second one. How about that?
Okay.
So what we've done so far is we've used a distribution to feed into the Monte Carlo, and we've kind of taken the longitudinal changes as part of that distribution. Of course, it would be feasible if we had more data to populate that, to improve the model to include some of those temporal variability, but we just haven't yet because the shedding data is so sparse. But it's definitely an important consideration because we know that the shedding is going to change over the course of infection.
Yeah, and basically, like, if we have a distribution for each of the time, like duration after infection, for example, you can then sort of average it out, and based on that, you can use that overall distribution to make predictions. And based on changes of that, you can then, so our model is also powerful because it's very flexible, like, and adaptable to whatever change of input value you use. So
Thank you.
yeah.
Do you have a sense for how immunity and vaccination might affect shedding? Have you found in your lit reviews any information on that?
Not specifically for the targets that I was examining, and actually because since shedding already is very limited, a very important next question would be exactly to examine not only like how does immunity, or how does vaccination, but also like how does asymptomatic versus symptomatic compare, and incorporating all those shedding dynamics versus like whatever different situations will all based on like different behavior will all impact the model, and therefore make a more better accurate model prediction. But right now we just don't have that information, unfortunately, but that's definitely something that needs to be addressed in the future.
And it's something that we know matters, right? I mean, for example, Aaron Bivens, a former postdoc in our group who's now at LSU, led a paper here at Notre Dame sampling prior to and post to required vaccination with higher than 95% compliance. And we saw the signal of SARS-CoV-2 completely drop out. So we know it matters. We just don't have a good way of quantifying it yet.
Yeah, there's a few related questions on shedding that have come in, and I think you've covered all of them. I guess you've just said there's a lot of uncertainty. Going back to the mpox data that you showed on shedding, can you remind us how many patients those that data came from when you showed the mpox shedding data?
Actually, the mpox data came from animal data from like chimpanzees, which is our closest relative. But we have like around 50 data points for Zika virus, though. We have a lot more data, and it's from actual Zika patients, like over 200 urine sample data. So yeah, I hope that kind of answers the question, at least.
And one interesting thing that's come up with monkeypox because of the skin lesions is we don't account for any other shedding routes here. So, for example, from skin in a shower or something like that. And so it's possible that we're missing shedding routes into the wastewater as well. So that's another consideration to think about with regards to shedding.
Yeah, so this is like a conservative estimate. So even if at the worst conservative case, it's saying that it's detectable, then that means it's definitely going to be detectable. If, like what Kyle mentioned, if skin even plays an additional adding to the concentration.
Great, thanks. I don't think you mentioned very much about the assumptions you made about like fate processes affecting virus deposited into the waste stream, such as decay or sorption. Can you talk about what your model considers for those processes?
So for persistence, we can factor that in if we know the decay rate and the time from toilet to the wastewater treatment facility. As for particles, that's also something that we can account for if we know, for example, the equilibrium partitioning between the liquid medium versus the solid medium. And then we can make a separate model for just the wastewater liquid portion versus the solid portion. But yeah, that's something that needs to also be addressed for each specific target, because every target is going to have a different behavior.
Thank you. And do you have this model available for people to use if they'd like to, or is there a package for it?
There's not a software package. We recently published this in Water Research. I'm sure if folks are thinking about how to do this, William would be glad to answer any questions, but it's detailed in these papers and in Water Research, but it's not like a comprehensive R package or Python package or something.
Yeah, if you just follow this procedure, you can do this for any disease. The key is you need that shedding data. If you don't have that, you can't apply this model. So yeah.
There's a few more questions I think we can get through. What sort of a specific question about the model is, do you account for underreporting when using case reports for your input for your model?
That's a very great question. And if we, for example, know the sort of a correction factor, like an estimation, like I know there's a study on malaria, for example, that sort of predicts based on a mathematical model, like the case rate that's reported is underreported by say 200-fold, then we can basically, so like for example, this plot, if we know that this is based on reported case rate. So if we factor in like 100-fold, then everything here would shift to the right by 2 units of magnitude, orders of magnitude, and that would therefore do something like this, like a shift, which means it will be a lot more feasible if we account for the correction in underreporting. And that again is something that needs to be sort of better understood to make more accurate model predictions. But again, our model is based on a conservative estimate of the worst case scenario where the reported case rate equals the actual, but that of course is not likely the case for many diseases, especially like Zika virus, which was mentioned, like 80% of them are asymptomatic. So
thanks.
Hope that answers the question.
There's also a question about how this might relate to sequencing. Have you thought about that at all?
No, I mean, I think the big gap there would be the quantitative nature of sequencing, and sort of the suitability of a sequencing for a target has a lot to do with other contributors to the nucleic acid pool. So it's not just the concentration of your target, but just how much DNA do you have there. But no, we haven't really thought about that, but that'd be interesting to consider.
Thanks. And then the last question we have here, can you explain again why your model predicts that you need a lower number of replicates in countries outside the United States? What is the cause of that result?
So yeah, that great question, and I wish I elaborated more on it, but it's basically comes down to 2 factors when it comes to location: the per capita wastewater flow rate and the relative case rates. So in the example for Zika, the reason why Brazil and Colombia have much lower requirement for the PCR replicate versus the US is because the lower per capita wastewater flow, like Brazil is 34% out of the US, Colombia is like 3%. But I think even more importantly, it's simply because you expect a lot more cases in endemic countries like Brazil and Colombia versus the US, where it's like few cases per year from travelers. So you probably would pretty much forget about trying to apply this in the US, unless in the future, somehow, due to climate change or whatever, Zika cases spike like 1,000-fold or something, then that's a different scenario. But yeah, I hope that answers the question.
Okay, we're out of time, everyone. Thank you. Have a very happy holidays, and we'll see you in February. And thank you to our speakers who did a great job today.
Thank you, everybody.
Thanks.
Bye.